Welcome to Fluent Daily, the English listening channel for advanced learners at B2 and C1 level. Every episode features a rich, flowing conversation in natural English on a topic that is genuinely worth thinking about: history, psychology, science, philosophy, and more. There are no simplified sentences here, no slow speech, no vocabulary drills. Just real English spoken naturally on ideas that matter. This is Fluent Daily. Let's get into it.
Today we have a great story, so let's get started right now. 100,000 people will die today from a single universally shared terminal illness.
Yeah.
And it's a condition that every single one of us listening right now currently has. Like, it is slowly progressing in your body as we speak.
It's a sobering thought, really.
It is. And if a new virus emerged tomorrow that killed 100,000 people every 24 hours, I mean, we would shut down the global economy.
Oh, absolutely.
We'd mobilize every scientist on Earth. We'd spare absolutely no experts to cure it. But because this particular condition is something we've dealt with since the, dawn of humanity, we just sort of accept it.
Right. There's this deep cultural fatalism about it.
Exactly. We call it natural. But today we are talking about the disease of aging. And more importantly, we are exploring the very real, very cutting-edge science that is actively trying to cure it.
Which is arguably the most profound biological paradox we face. We accept aging as this inevitable fact of life, like gravity.
Yeah, you're born, you grow, you peak, you decline, and you die.
Right. But when you look at it through the lens of pure molecular biology, aging isn't some mystical, unstoppable force.
It's just biology, right?
Exactly. It's a terrifyingly complex, but ultimately mechanical cascade of microscopic failures. And theoretically, well, those failures don't actually have to happen.
Which is just wild to think about. And that is exactly our mission for this deep dive. We are throwing out all the platitudes about, aging gracefully.
Yeah, we're going much deeper than that.
We really are. We are going to look under the hood of human biology to understand the exact mechanisms of your physical decline.
The nuts and bolts of it.
Right. We're going to look at the radical therapies, some of which are available right now, and some that are still in the lab that are trying to slow, stop, or literally reverse that decline.
And, of course, we have to talk about the fallout of all this.
Oh, the ethics. Absolutely. Because if we actually figure out how to double human life expectancy, what happens to society?
What happens to human psychology? It's a huge question.
It is. But before we get into the mechanisms and the ethics, I think it's crucial to define what the scientific community is actually trying to achieve here.
Yeah, that's a really important starting point. Because there's a very common misconception that gerontology, which is the study of aging, is just about extending lifespan.
Right, and nobody wants that. I mean, nobody wants to be kept alive on a ventilator for an extra 40 years.
No, exactly. Just dragging out the most miserable part of life.
Yeah, that sounds like a nightmare.
It does. And that's why lifespan is just the chronological clock. It's just the total years you're breathing. The actual gold standard in modern longevity research is something called health span.
Health span. Okay, explain the difference for the listener.
So health span is the period of your life spent in good health. It's about maintaining functional independence, cognitive sharpness, vitality.
So being able to actually do things.
Right. The objective is to compress the period of sickness, what we call morbidity in the field, into the absolute shortest possible window right at the very end of a very long life.
Okay, so the goal is to have you playing tennis, traveling, and living fully at 110.
Yes.
Yeah.
And then essentially just powering down quickly when the time comes, rather than a decades-long decline.
I love that framing, health span over lifespan. But to understand how to extend that health span, we really have to start with the most basic, honestly baffling question.
Why do we age at all?
Exactly. Why do we age? Because if you think about natural selection, the whole point is survival of the fittest.
Right.
You would assume that evolution would heavily favor an organism that lives a really, really long time, because a longer life means more opportunities to reproduce and pass on your genes.
It's a brilliant question. And honestly, it stumped evolutionary biologists for decades.
It just seems counterintuitive.
It does.
Mm-hmm.
But you have to remember, we didn't evolve in a world with antibiotics, indoor plumbing, and grocery stores.
No, we evolved in a pretty brutal environment.
Incredibly hostile. So in the 1950s, an evolutionary biologist named Peter Medawar proposed what he called the mutation accumulation theory.
Mutation accumulation.
Okay. Medawar pointed out that in the ancestral environment, Almost no one survived to old age anyway.
Because of the environment.
Right. Most humans, and most animals for that matter, died from predation, accidents, or infectious diseases before they even reached the age of 30 or 40.
Starvation, freezing winters, massive infections from tiny cuts. So the average life expectancy was just incredibly short due to external factors.
Exactly. So look at this from the perspective of natural selection.
Okay.
Natural selection works by weeding out genetic mutations that harm an organism before it can reproduce.
Because if a gene kills you at age 10, you don't have kids, so it never gets passed on.
You've got it. But what if there's a genetic mutation that causes a fatal heart defect or a virulent cancer, but it only activates at age 70?
Oh, I see. Well, in the ancestral world, you're already dead at 40 anyway from a saber-toothed tiger or an infection.
Exactly. So that gene never gets weeded out. Natural selection is effectively blind to genetic variants that cause damage late in life.
Because those individuals have already reproduced at age 20.
Right. The harmful gene is passed on before the negative effects ever materialize. The evolutionary pressure to maintain the body drops to near zero after your primary reproductive years.
So the mutations just accumulate over generations in the human genome. They're just hiding in the shadows of old age.
Precisely. That's Metawar's theory.
Okay, that makes total sense. Evolution just stops paying attention to us after we have kids because we were probably going to get eaten anyway.
Basically, yes.
But there's another layer to this that I found absolutely fascinating when we were looking through the research. It's George Williams's antagonistic pleiotropy theory.
Oh, yes. This one is really interesting.
And I want to make sure I understand this one because it sounds like evolution is actively sabotaging us, not just ignoring us like Metawar suggested.
It is a much more active and honestly somewhat sinister mechanism.
Okay, lay it on me.
So pleiotropy is a genetic term. It means that a single gene controls more than one distinct trait.
Okay.
And antagonistic means those traits work against each other.
Right. So let me try an analogy here to see if I'm tracking the actual biological reality.
Go for it.
Imagine you are an engineer, right? And you're designing a race car.
Okay.
Your only goal is to win the very first 10 races of the season. So you install this massive experimental turbo boost system in the engine.
A huge advantage early on.
Right. It makes the car incredibly fast. You absolutely dominate those early races. But the extreme heat from that turbo boost guarantees that by race number 15, the engine block is going to warp, calcify, and completely destroy the car.
That is a brilliant analogy.
So in the wild, evolution is the engineer, and it takes that deal every single time, right?
Every single time. That is a perfect translation of the biology. Evolution always prioritizes early life reproductive fitness over late life survival. Always.
Because again, it assumes you won't make it to late life.
Exactly. So let's look at a biological example of that turbo boost.
Yeah.
Think about cellular senescence, which we are going to get into deeply later on.
Oh yeah, the zombie cells.
Right. So the ability of a cell to permanently stop dividing is actually a crucial defense against cancer when you were young.
Oh, so it's a tumor suppression mechanism.
Yes. That's the early life benefit. The turbo boost, it stops a mutated cell from becoming a tumor so you can live long enough to have kids.
Okay, but what happens later?
Well, that exact same mechanism, operating decades later, causes a massive buildup of these arrested zombie cells.
And they cause problems.
Huge problems. They secrete inflammatory chemicals, which drives tissue aging and chronic disease.
Wow. So the mechanism that saved my life at 20 is the exact same mechanism that degrades my tissues at 70.
Yes. Another classic example is calcium metabolism.
How does that one work?
Well, when you are young, you need to extract calcium from your blood and deposit it really aggressively into your bones.
Right, so you can grow strong, run fast, survive.
That's the turbo boost again. But as you age, the regulatory systems around that aggressive calcium transport begin to falter.
Oh no, where does the calcium go?
It starts getting deposited in the soft tissues, particularly the walls of your arteries.
So the same genetic drive that gave me strong bones at 20 gives me calcified, hardened arteries at 80.
Exactly. The turbo boost burns out the engine.
Man. So aging isn't some glitch or a failure of evolution. It is a direct, calculated byproduct of evolution, prioritizing our youth.
Yes. And this perfectly aligns with a third theory developed by Thomas Kirkwood in the late 1970s. It's called the disposable soma theory.
Disposable soma. Soma meaning body.
Yes. Soma simply means the body, as distinct from the germline.
The germline being the sperm and eggs that carry our DNA to the next generation.
Right. So Kirkwood looked at aging as an energy allocation problem.
Okay, like a budget.
Exactly. Organisms have a finite amount of energy derived from food. So evolution has to make this cold mathematical choice.
What's the choice?
Do we invest this limited energy into perfect bodily maintenance like flawless DNA repair, indestructible proteins, or do we invest it into rapid growth and reproduction?
And obviously reproduction wins.
It has to, or the species dies out. Perfect maintenance is incredibly energy intensive. So evolution basically builds us out of biological cheap parts.
Wow. Cheap parts.
Yeah, it gives us just enough DNA repair capacity to reach sexual maturity and raise offspring.
And once the genetic cargo is delivered.
The vessel, the soma, is entirely disposable. There is zero evolutionary return on investment for keeping a 70-year-old body perfectly maintained.
I have to say, taking this macro view is a little insulting. It implies my entire physical existence is just cheap packaging for my DNA.
From a purely evolutionary standpoint, I mean, it is.
That is bleak, but it also sets the stage perfectly. So to summarize the hand we've been dealt. Natural selection is blind to late-life diseases. It actively installs biological turbo boosts that eventually destroy our tissues. And it refuses to invest energy in our long-term repair because it assumes we're disposable packaging.
That's a very accurate summary.
Okay, so if that is why we fall apart, the next logical question is how? What is the actual physical machinery of this breakdown?
Right. And for a very long time, answering that question was like the old parable of the blind men and the elephant.
Everyone is feeling a different part.
Exactly. The field of aging research was incredibly fragmented. You had geneticists studying DNA damage, cell biologists looking at mitochondria, biochemists studying proteins.
All looking at different parts of the same elephant?
Yes. But in 2013, a group of scientists led by Carlos Lopez Otin published this landmark paper in the journal Cell.
This was a huge deal, right?
Oh, it was a massive attempt to unify the entire field. They categorized the biological breakdown of aging into 9 distinct cellular and molecular processes.
And they called these the hallmarks of aging.
Yes. It essentially became the periodic table for gerontologists.
Right. It gave everyone a shared vocabulary.
Exactly. They actually updated it in 2023 to include 13 hallmarks, recognizing new discoveries like microbiome dysbiosis and chronic inflammation.
Okay, but looking at the original core hallmarks is probably the best way to understand the physical reality of aging, right?
Definitely. It provides the best foundational framework.
Okay, let's walk through this. But instead of just reading down the list like a textbook index, I really want to understand how these things connect.
Okay, the domino effect.
Yeah. How does a single human cell go from young and healthy to old and broken? Where does the very first domino fall?
The first domino almost always falls at the level of your biological blueprint. The first hallmark is genomic instability.
So DNA damage.
Yes. And people think of their DNA as this static protected library. But it's not. It is under constant, relentless assault. Every single day, the DNA in every cell of your body experiences tens of thousands of lesions.
Tens of thousands from what?
From everything, really. From ultraviolet radiation from the sun, from environmental toxins, X-rays.
So mostly from the inside, right?
Yes, mostly from your own metabolism.
Yeah.
Specifically, reactive oxygen species created when your cells generate energy, they constantly bombard your DNA.
So my own body's engine is producing the exhaust that damages my own blueprint.
That's a great way to put it. They cause chemical modifications, single-strand breaks, and the most dangerous of all, double-strand breaks where the DNA helix is completely severed.
But our bodies have repair mechanisms for that, don't they?
We do. We have incredibly sophisticated DNA repair machinery. Proteins that constantly patrol the genome, snipping out damaged bases and patching the holes.
But let me guess, disposable soma theory.
You got it.
Yeah.
Evolution didn't give us perfect repair machinery. It gave us good enough machinery.
Right. So over decades, errors just slip through.
Exactly. The repair proteins might patch a break incorrectly, leading to a mutation. Over time, this genomic instability acts like static accumulating on a radio broadcast. The instructions for how the cell should behave get corrupted.
Okay, so the core code gets corrupted. But what about the physical structure of the chromosomes themselves? Because I know that's another major hallmark, telomere attrition.
Yes, telomeres.
We hear about telomeres all the time in anti-aging articles. How do they fit into this?
Telomeres are intimately connected to genomic instability. Think of your chromosomes, the long strands of DNA like shoelaces.
Okay.
Telomeres are the little plastic tips, the aglets, at the very ends of the shoelaces. Their job is to protect the ends of the DNA from fraying or from fusing with other chromosomes.
But they don't last forever.
No, they don't. And this goes back to the 1960s, to a scientist named Leonard Hayflick. He discovered that normal human cells cannot divide indefinitely in a petri dish.
Right, they hit a wall.
Yes, they divide about 50 times, and then they permanently stop. This is known as the Hayflick limit.
And the reason for this limit is the telomeres wearing down.
Exactly. Every time a cell divides, its DNA must be copied. But because of a quirk in the way the copying machinery works, it can't copy the very tip of the chromosome.
So with every division, the plastic tip on the shoelace gets a little bit shorter?
Yes. And when that telomere gets critically short, the cell senses it as massive DNA damage.
Oh, so the cell thinks its genome is literally unraveling.
Right. So to prevent itself from turning into a chaotic, cancerous mass, it hits the emergency brake. It triggers another hallmark, cellular senescence. It permanently stops dividing.
Wow. Okay, so the genomic instability and the shrinking telomeres trigger the cell to shut down.
Yes.
But wait, if the DNA is getting corrupted with mutations before it shuts down, does the cell start acting weird? Like, does it start misreading the instructions?
It does. And that leads us to a crucial hallmark called epigenetic alterations.
Epigenetics. I want to spend time on this because it's so central to the therapies we're going to discuss later on.
It really is.
So if the DNA is the hardware, the epigenome is the software, right?
That is a very accurate way to think about it. Every cell in your body, a skin cell, a heart cell, a neuron, contains the exact same DNA, the exact same instruction manual.
So what makes a heart cell, and a skin cell, a skin cell?
It's the epigenome. It's a complex system of chemical tags like methyl groups that attach to the DNA.
What do the tags do?
They act like highlighters and redacting tape. They tell the heart cell to read the heart genes and ignore the skin genes.
So it basically dictates which genes are turned on and which are turned off.
Precisely. But as we age, and as genomic instability accumulates, this epigenetic software gets corrupted.
The scratches on the CD.
Right. The tags fall off where they should be attached,
Yeah.
and attach where they shouldn't be. The strict boundaries that define a cell's identity start to blur.
So what happens? A skin cell might start inappropriately turning on genes that belong to a liver cell.
Exactly. The cell is losing its identity. It's forgetting how to do its specific job because the software is glitching.
And when the software glitches, the physical products of the cell also start to fail, I assume.
They do. Which brings us to the loss of proteostasis.
Proteostasis.
Yeah.
Proteo for proteins, stasis for stability or balance.
Right. Proteins are the microscopic workhorses of your body. They build tissues, carry oxygen, digest food, fight infection.
They do everything.
But to work properly, a protein must be folded into a highly specific three-dimensional shape. If it's misfolded, it's useless and often dangerous.
Dangerous how?
Well, your cells have a quality control system, including helper molecules called chaperone proteins, that assist in folding and clear away the garbage.
Okay.
But as the epigenetic software corrupts and energy levels drop, this quality control network crashes. Misfolded proteins start clumping together inside the cell.
Wait, is this what happens in Alzheimer's disease?
It is the exact mechanism. In Alzheimer's, misfolded amyloid and tau proteins aggregate in the brain because the neuron's proteostasis networks have failed.
Wow. So you really see how the dominoes fall.
You do. DNA damage and telomere shortening cause epigenetic software glitches.
The glitches cause the protein factories to produce misfolded junk.
The junk accumulates and triggers cellular senescence.
And as I recall, those senescent cells, which we are definitely going to talk about, start secreting inflammatory chemicals, which then damage the surrounding cells, creating even more genomic instability.
It is a self-amplifying downward spiral. The hallmarks are not isolated silos. They are an interconnected web of failure.
Which sounds terrifying. But there's a silver lining here, isn't there? I mean, the whole reason Lopez Otin and his team mapped this out wasn't just to depress us.
No, not at all. By defining the specific mechanisms, they gave science a target list.
That is the paradigm shift right there.
It is. Before this framework, aging was this abstract, unbeatable phantom. You couldn't treat aging. You could only wait for a heart attack and try to treat the heart attack, or wait for cancer and treat the cancer.
But the hallmarks of aging turned aging into a solvable engineering problem.
Exactly. Because if we can intervene at the fundamental level of the hallmarks, we aren't just treating one disease, we're treating the root cause of all of them.
Right. If we can stabilize the genome or fix the epigenetic software or clear out the misfolded proteins,
We could theoretically prevent or delay the heart attack, the cancer, and the Alzheimer's all at the same time. We shift from a reactive medical model to a proactive, preventative one.
Okay, so let's talk about actually intervening. Let's look at the interventions. Because out of all the hallmarks, there is one that seems to hold the key to the most proven, most robust anti-aging intervention we have ever discovered.
Yes.
And I'm talking about the hallmark called deregulated nutrient sensing, how our cells process food.
If you want to understand how to manipulate lifespan, you have to look at how organisms respond to scarcity.
Wait. You're telling me that the most scientifically validated way to live longer involves starving myself?
Well,
Because I'm going to be honest, that sounds like a miserable way to get an extra 10 years. If I can't eat a burger, what's the point?
it is a bitter pill to swallow, no pun intended.
Right.
But biologically, it is undeniable. We have known this since the 1930s.
In the 1930s.
Really? Yes. A nutritionist named Clive McKay at Cornell University ran a series of experiments on rats. He gave one group of rats as much food as they wanted. They lived typical rat lifespans. He took another group and restricted their caloric intake by about 30%. But crucially, making sure they still got all their essential vitamins and minerals so they didn't suffer from malnutrition.
Right. So undernutrition without malnutrition.
Exactly. And the results were staggering. The calorie-restricted rats didn't just live a little bit longer, they lived dramatically longer. Their maximum lifespans were extended by 30 to 40%. They stayed youthful, energetic, and disease-free long after the control rats had died.
That's huge.
It is. And this wasn't a fluke. In the nearly 90 years since McKay's experiment, this finding has been replicated across the entire tree of life.
Really? Not just in rats.
If you restrict calories by 20 to 40% in yeast, in worms, in fruit flies, in mice, and even in non-human primates like rhesus monkeys.
You see significant extensions in health span and lifespan.
Yes, every time.
30 to 40%. I mean, if you map that onto a human life, that's like taking an 80-year lifespan and pushing it out to 110 or 120 just by changing your food intake.
Biologically, yes.
But how? What is the actual mechanism? Why does depriving the body of energy make it live longer? You'd think starvation would cause the system to break down faster.
To understand the how, we have to zoom deep inside the cell to a specific protein complex called mTOR. mTOR.
Okay.
That stands for the mechanistic target of rapamycin. mTOR is essentially the cell's master nutrient sensor.
Like a thermostat, but instead of sensing temperature, it senses food.
That's a great analogy. Specifically, it senses amino acids and energy levels. It basically asks the question, is there abundant food in the environment?
Okay, so when I eat a heavy meal,
Especially one rich in protein, amino acids flood your bloodstream. mTOR senses this abundance and turns on.
and what does on mean for the cell?
When mTOR is, it signals the cell to grow. It says, we have plenty of building blocks. Let's synthesize new proteins, let's divide, let's build new tissue.
Which is great when you were young and growing, or if you were a bodybuilder trying to put on muscle.
Yes, exactly. But remember, antagonistic pleiotropy.
Oh, right. The turbo boost.
High mTOR activity late in life drives unregulated cellular growth, which is a key driver of cancer and accelerated aging.
Okay, so what happens when you restrict calories? What happens when food is scarce?
The mTOR thermostat turns off.
The cell senses scarcity.
Exactly. And when mTOR is inhibited, it triggers an incredibly elegant survival mechanism called autophagy.
Autophagy, let me break down that word. I know auto means self, and phagy means eating, so self-eating.
Yes. When the cell realizes it doesn't have an influx of new energy coming from the outside, it has to find energy on the inside.
So it eats itself?
Parts of itself. It starts looking around for cellular garbage. It finds misfolded proteins, damaged mitochondria, and oxidized lipids.
The stuff that causes the hallmarks of aging.
Right. It builds a double membrane structure called an autophagosome, which is literally a microscopic garbage bag, and it wraps up all the cellular trash.
Okay, it bags the trash, then what?
It delivers that garbage bag to a structure called the lysosome, which is basically a cellular incinerator filled with acid and digestive enzymes.
Wow.
The lysosome breaks the garbage down into its basic molecular building blocks and recycles them for new energy.
That is incredible. Let me run an analogy by you because I use this to explain autophagy to people and I want to make sure the biology holds up.
Let's hear it.
Think of a startup company. When the company just lands a massive $100 million round of venture capital funding, which is like having abundant food and high mTOR, right?
Right.
Management expands recklessly. They hire tons of people, they buy fancy office furniture, they rent huge office spaces. They aren't paying attention to efficiency at all. Growth at all costs is the only goal.
A very apt comparison for an overfed biological system.
But then a severe recession hits. The funding dries up completely. That's caloric restriction. The MTOR signal drops.
Yeah.
What does management do? They can't grow anymore, so they're forced to look inward to survive. They implement autophagy. They fire the underperforming employees. They sell the fancy furniture. They consolidate their resources, and they clean up their accounting books.
They get lean.
The company becomes lean, highly efficient, and optimized for survival.
That is fundamentally what is happening in your cells. By artificially inducing a state of scarcity through caloric restriction, you force your cells to clean house.
You are constantly clearing out the damage.
You are constantly stimulating the clearance of the very damaged, the misfolded proteins, the broken mitochondria that drives the hallmarks of aging.
It makes total sense. But we have to be real here. Asking the average person to cut their caloric intake by 30% for their entire adult life is a complete non-starter. Our entire culture is built around food. It's socially isolating, it leaves you feeling cold and tired, and frankly, people just love pizza too much. The compliance rate would be 0.
You are entirely correct. True caloric restriction is psychologically grueling for humans, which is why the holy grail of gerontology right now is finding a pharmacological way to mimic this process.
Can we trick the body into thinking it's starving and trigger that cellular house cleaning without actually restricting calories? We want the autophagy without the misery. Give me the pill.
Well, the astonishing thing is we already have it.
Wait, really?
The clue is right there in the name of the nutrient sensor, the mechanistic target of rapamycin.
Rapamycin. I've heard of this. But isn't this an anti-rejection drug for organ transplants? How does an organ transplant drug fit into life extension?
The story of rapamycin is one of the greatest accidental discoveries in medical history. In 1964, a team of scientists went to Easter Island, the indigenous name for which is Rapa Nui. They were taking soil samples, looking for new antimicrobial compounds. They found a specific bacterium that secreted a chemical weapon to kill neighboring fungi.
Oh, wow.
They isolated this chemical, named it rapamycin after the island, and found it was a potent immunosuppressant. So it was developed to stop the immune system from attacking transplanted kidneys.
Okay, so it suppresses the immune system. That still doesn't explain the anti-aging part.
Fast forward decades later, biologists are studying this drug, and they realize how it suppresses the immune system. It works by physically binding to a protein complex, and that binding strongly inhibits the MTOR pathway.
Oh, it tricks the cells into thinking they are starving.
It flips the thermostat off artificially.
Yes. So in 2009, researchers in the Interventions Testing Program, which is a rigorous multi-lab initiative funded by the NIH, decided to test rapamycin on mice.
And the results?
Yeah.
Unprecedented. Rapamycin extended the lifespan of both male and female mice significantly. And what was truly remarkable, they started feeding the drug to the mice when they were 600 days old.
Which, in human terms, is what?
It is like starting the treatment at age 60.
Wait, so they didn't have to be on it their whole lives. You could take an old mouse, give it rapamycin, trigger the autophagy, and it still extended its lifespan.
Yes. It showed that pharmacological intervention late in life could still dramatically delay aging.
Okay, I am going to play devil's advocate here because this sounds way too good to be true.
Please do.
You said rapamycin is a potent immunosuppressant used for organ transplants. If I start taking rapamycin to live to be 120, aren't I going to die of pneumonia next week because my immune system is completely shut down?
That is the exact translational hurdle we are facing right now. Daily high-dose rapamycin will absolutely suppress your immune system and make you vulnerable to infections.
Right.
However, the nuance lies in how MTOR is structured. There are actually two distinct MTOR complexes in the cell, MTOR-C1 and MTOR-C2.
Okay, what's the difference between the two?
Broadly speaking, inhibiting MTOR-C1 gives you the lifespan extension and the autophagy. Inhibiting MTOR-C2 leads to the negative side effects, like immune suppression and insulin resistance.
Ah, I see.
The prevailing theory right now is that continuous daily dosing inhibits both. But intermittent dosing, say, taking a specific dose of rapamycin once a week, might selectively inhibit MTOR-C1 just long enough to trigger the cellular house cleaning, but allow MTOR-C2 to recover, keeping the immune system intact.
Ah, so it's like doing a deep clean of the house once a week rather than trying to clean it 24/7.
Exactly. There are ongoing human clinical trials right now looking at intermittent dosing of rapamycin and newly developed rapalogs, analogs of the drug designed to be safer, to see if we can capture the longevity benefits without the immune compromise.
That is fascinating. So we have a potential pathway to slow down the accumulation of new damage. But here is my concern. Slowing down growth and clearing out some cellular garbage is great for maintenance. But what about the heavy damage that has already accumulated? Let's say I'm 60 years old. I've got decades of DNA damage built up in my tissues.
Right.
Caloric restriction isn't going to fix a broken cell. What do we do about the fundamentally corrupted cells that are already lingering in the body?
That brings us to one of the most dynamic and rapidly moving areas in all of gerontology. It's time we talk in depth about cellular senescence.
Okay, here's where it gets really interesting. In our last discussion, we briefly touched on this, but let's go all the way in. We are talking about zombie cells.
Zombie cells is the perfect colloquial term. Let's go back to our hallmarks of aging.
Okay.
When a cell experiences severe stress, maybe its telomeres get too short or its DNA suffers a double-strand break from radiation, it faces a choice. If it keeps dividing with broken DNA, it will become cancerous.
Right.
So, as a highly evolved tumor suppression mechanism, it hits the emergency brake. It enters senescence. It permanently, irreversibly stops dividing.
Okay, so hitting the brakes on cancer is obviously good. I want my damaged cells to stop dividing. So what is the problem? Why are they zombies?
Because they don't die. Ideally, a severely damaged cell should undergo apoptosis-programmed cell death. It should cleanly dismantle itself so the immune system can sweep it away.
But they don't do that.
No, senescent cells resist apoptosis. They linger in the tissues. They aren't dead, but they aren't fully functioning healthy cells either. They're in a twilight state.
Thus, the zombie moniker.
Yes. And the real danger isn't just that they are taking up space, it's what they're actively doing while they sit there. Senescent cells undergo a massive shift in their gene expression, and they begin to secrete a highly toxic cocktail of chemicals called the senescence-associated secretory phenotype, or SAP.
The SASP. What exactly is in this toxic cocktail?
It's a vicious mix of pro-inflammatory cytokines, chemokines, and tissue-degrading enzymes called proteases.
So they are essentially spewing out biological acid and inflammation signals into the surrounding healthy tissue. It's like having a rotten apple in a barrel that actively starts rotting all the other apples around it.
That is exactly what happens. The SAP causes chronic low-grade inflammation throughout the body. We call it inflammaging.
Inflammaging.
And this inflammation damages neighboring healthy cells, often triggering them to become senescent as well. It's contagious.
Wow, so the zombies bite the healthy cells.
Pretty much. When you were young, your immune system is usually robust enough to hunt down and clear out these senescent cells. But as you age, the immune system weakens, and the rate of damage increases.
So the zombies just accumulate.
They do. They build up in your joints, causing osteoarthritis, in your blood vessels, driving atherosclerosis, in your brain, contributing to neurodegeneration.
So if these zombie cells are driving all these different diseases, the solution seems incredibly obvious. We just need to kill them.
That's the idea.
But how do we hunt down a zombie cell without killing the healthy cells next to it? In a previous conversation, we used the analogy of a cellular shotgun, but I feel like that's wrong. A shotgun would destroy the healthy tissue too. We need something highly targeted.
You are right. A shotgun is the wrong analogy.
Yeah.
It's much more like a guided missile targeting a very specific defense shield. Enter a new class of drugs called senolytics.
Seno for senescence. Senelic meaning to destroy.
Exactly. The challenge was exactly what you described. How do you kill a senescent cell without harming a healthy one?
Right, because chemotherapy kills cancer cells, but it's brutally toxic to healthy dividing cells.
Exactly. Researchers, particularly groups at the Mayo Clinic and the Scripps Research Institute, asked a brilliant question. If a senescent cell is so damaged, why doesn't it just undergo apoptosis and die naturally?
Right, why doesn't it just self-destruct?
They discovered that senescent cells survive by upregulating specific anti-apoptotic pathways. They create a biochemical shield that blocks the self-destruct signal. We call these SKP networks, senescent cell, anti-apoptotic pathways.
So the zombie is actively holding a shield up to stop itself from dying.
Yes, so senolytic drugs are designed to specifically target and disable that shield. If you knock out the SKP network, the senescent cell, which is already primed to die because of its massive internal damage, simply collapses and undergoes apoptosis.
Oh, wow.
The healthy cells, which don't rely on those emergency shields, are left completely unharmed.
That is incredibly elegant. And this isn't just theoretical mouse biology anymore, is it? We have actual human trials testing this.
We do. and the early results are a proof of concept for a new era of medicine. In 2019, Dr. James Kirkland and his team at the Mayo Clinic published the results of a first-in-human trial using senolytics.
What did they use?
They use a combination of two drugs. The first is doximib, which is an FDA-approved chemotherapy drug used for leukemia.
Okay.
The second is quercetin, which is a naturally occurring flavonoid, a plant pigment found in apples, onions, and capers.
That is a wild combination. A heavy-duty leukemia drug and an extract from an onion. Why those two?
Because through extensive screening, they found that dasatinib disables one specific set of anti-apoptotic shields, and quercetin disables another. Together, they create a broad-spectrum senolytic capable of clearing multiple types of zombie cells.
Who did they test it on?
Kirkland's team tested this combination on patients suffering from idiopathic pulmonary fibrosis.
What is that?
It is a devastating progressive lung disease where the lung tissue becomes thick and scarred. It is highly associated with aging, and crucially, lung biopsies from these patients show a massive burden of senescent cells actively secreting tissue-destroying SSP factors.
So the zombie cells are literally scarring the lungs from the inside out. Did the drugs work?
The trial was small, primarily designed to test safety, but the results were striking. After just a few intermittent doses of the dasatinib and quercetin combination, the patients showed significant, measurable improvements in their physical function.
Like what?
They could walk further distances and stand up from a chair faster. The senolytic treatment successfully reduced the burden of senescent cells and alleviated the physical symptoms of the disease.
If we pull back and look at the bigger picture here, this fundamentally changes how we think about medicine.
Completely.
Right now, if I go to the doctor as a 70-year-old, it's a game of whack-a-mole. I have high blood pressure, so I get a beta blocker. My knees hurt from osteoarthritis, so I get an anti-inflammatory. My blood sugar is creeping up, so I get metformin. I'm taking 5 different pills for 5 different symptoms.
Treating the end-stage diseases of aging in isolation.
But as senescent cells and their toxic SS are the underlying fire driving the blood pressure, the joint degradation, and the insulin resistance, taking a senolytic could theoretically extinguish the fire everywhere. One therapy could treat multiple age-associated conditions simultaneously.
You kill the zombies and your lungs breathe easier, your joints stop hurting, and your vascular system relaxes. That is the ultimate promise of xenotherapeutics. It is the shift from treating symptoms to treating the root mechanism. And the field is exploding. There are dozens of biotechnology companies currently in clinical trials with next-generation senolytics, trying to perfect the targeting and minimize side effects.
It is incredibly hopeful. Okay, so let's review our arsenal. We can slow down the accumulation of new damage by tricking the mTOR pathway with caloric restriction or rapamycin.
Yes.
We can go in and clear out the heavily damaged inflammatory zombie cells using senolytics. This is amazing maintenance, but it still feels like we are just slowing the ship from sinking.
Well, slowing it down is a good start.
True, but can we actually hit undo? Can we take an old, exhausted, functioning cell and somehow wind the clock backward? Can we make an old cell genuinely young again?
This brings us to the bleeding edge. What you are asking about is arguably the most conceptually radical, paradigm-shattering idea in all of biology, epigenetic reprogramming.
Okay, strap in. Reversing the biological clock. How is this even theoretically possible?
To understand this, we have to travel back to 2006, to the laboratory of a Japanese researcher named Shinya Yamanaka. What Yamanaka discovered was so profound, it won him the Nobel Prize just 6 years later.
What was he studying?
He was studying stem cells. Remember, an embryonic stem cell is pluripotent. It has the potential to become any cell in the body. A skin cell, a neuron, a liver cell.
Right, and as the embryo develops, those cells differentiate. They get assigned a specific job.
Exactly. And the dogma in biology for decades was that differentiation was a one-way street. Once a cell became a skin cell, it was permanently a skin cell. Its fate was sealed.
Makes sense.
But Yamanaka hypothesized that the instructions to be a stem cell were still hidden in the DNA of adult cells, just locked away by epigenetic marks. He screened thousands of transcription factors, proteins that turn genes on and off.
He was looking for the master keys.
Yes, and he found them. He discovered that by introducing just 4 specific transcription factors into a fully adult, differentiated skin cell, he could completely erase its identity.
Just 4.
Just 4. These 4 master keys, OCT4, SOX2, KNLF4, and C-MYCAL, now famously known as the Yamanaka factors, wiped the epigenetic slate clean. The adult skin cell reverted entirely back into a pluripotent stem cell.
It literally traveled back in time biologically to an embryonic state.
Yes. It proved that the biological clock is not a one-way street. The information to be young is still in the cell.
That's incredible.
Now, while this is incredible for creating stem cells in a petri dish, you absolutely cannot do this in a living human being.
Well, why not? If I want young skin, why can't I just put some Yamanaka factors in a face cream?
Because if you fully reprogram a cell in vivo, in a living organism, it forgets what it's supposed to be doing. If the cells in your heart suddenly erase their identity and turn into stem cells, they stop beating.
Oh, right. That would be bad.
Furthermore, if you inject Yamanaka factors into an animal and leave them turned on, the cells rapidly divide and form massive, chaotic tumors called teratomas. So full reprogramming is a complete disaster for anti-aging.
So it's useless for living longer because it just gives you cancer and destroys your tissues.
That is what everyone thought.
Right.
Until researchers, most notably David Sinclair's laboratory at Harvard, asked a brilliant, nuanced question.
Which was?
What if we don't leave the Yamanaka factors on continuously? What if we just pulse them? What if we turn them on for just a few days and then quickly turn them off before the cell reaches the point of no return and forgets its identity?
A partial reprogramming.
Exactly.
Huh.
And what they discovered is astonishing. This transient pulse of the Yamanaka factors manages to wipe away the epigenetic marks of aging, the scratches that accumulate over time, but it stops just short of erasing the cell's core identity.
So a heart cell stays a heart cell.
Yes, but its biological age is completely reset to youthful parameters.
Okay, let me bring in my absolute favorite analogy for this, because Sinclair's information theory of aging is a total mind bender, and I want to make sure I'm explaining it correctly.
Okay.
Think of your DNA, your genome, like the digital music etched onto a CD. The music itself is the genetic code. When you are born, the CD is pristine, and the CD player's laser reads the music perfectly. The cells function beautifully.
The hardware is perfectly intact.
Right. But over decades of life, that CD gets dragged across the table. It gets scratched. Exposure to the sun, toxins, bad diet, radiation, the wear and tear of living. Those scratches represent the epigenetic alterations we discussed earlier. The methyl tags attaching to the wrong places.
Exactly.
Now, the music underneath, the underlying DNA sequence, is still perfectly intact. The code hasn't changed. But the scratches prevent the laser, the epigenetic reader, from reading the music. The cell starts skipping. It forgets how to function properly.
That is a brilliant visualization. It represents a profound shift in how gerontologists view aging. We used to think aging was primarily the permanent destruction of the hardware-mutated DNA that simply couldn't be fixed.
But it's not.
The information theory of aging argues that it's mostly a corruption of the software. The genetic code is still there, perfectly preserved, but the regulatory marks that control it have lost their place.
And so epigenetic reprogramming, that brief pulse of the Yamanaka factors, is essentially taking that scratched CD and running it through a polishing machine. It polishes out the scratches.
Beautifully put.
It doesn't change the music. It just allows the laser to read the original music perfectly again. The cell remembers how to be young.
And this isn't just theory. Sinclair's lab proved this in a groundbreaking paper published in Nature in 2020. They focused on the central nervous system, specifically the optic nerve of mice.
Okay.
Typically, if you crush the optic nerve of an adult mammal, it cannot regenerate. The neurons are old, they lack the epigenetic program for growth, and the mouse goes permanently blind.
So what did they do?
They used a viral vector to deliver 3 of the Yamanaka factors. They left out c-Myc, because it's a known oncogene that drives cancer, into the retinal ganglion cells of the mice. They turned the factors on after crushing the optic nerve. The transient reprogramming successfully reversed the epigenetic aging markers in the cells. The cells began to behave like embryonic neurons. They regenerated their axons, grew down the optic nerve to the brain, and functionally restored the animal's vision.
No way.
Yes. They made old blind mice see again by hitting reset on their epigenetic software.
That is genuinely staggering, reversing blindness by rewinding the age of the cells.
It is transformative. The implication is that if the epigenetic program of aging is resettable, then aging itself might be reversible in ways that simply targeting the downstream damage could never achieve.
But I have to push back here. I have to play the skeptic.
Of course.
Rejuvenating an optic nerve in a highly controlled, genetically modified mouse model is incredible science. But doing that to a human, you cannot just inject a virus-carrying Yamanaka factor systemically into a human body and hope for the best.
No, you absolutely cannot.
How do you ensure every single tissue gets exactly the right amount of polishing? If you over-polish a liver cell, it loses its identity and becomes a tumor. This sounds like a tightrope walk over a canyon of cancer.
Your skepticism is entirely warranted. This is the fiercest debate in the field right now. Applying partial epigenetic reprogramming systemically and safely in humans is a massive, currently unsolved hurdle.
It's just too risky right now.
It is a razor-thin therapeutic window, a little too much expression, and you induce teratomas and tissue dysplasia. Not enough, and you do nothing. While billions of dollars are flowing into biotech companies trying to solve this, we are a very long way from a systemic Benjamin Button pill.
Okay, so bringing this back down to earth. We've talked about evolutionary theory, we've talked about caloric restriction, rapamycin, zombie-killing senolytics, and futuristic epigenetic gene therapy.
A lot of high-level science.
It is incredible science, but what does this mean for the person listening to us right now? You cannot go to your local pharmacy and buy a bottle of Yamanaka factors.
Definitely not.
Rapamycin is difficult to get prescribed and potentially suppresses your immune system. So what actually works today? Let's translate this hardcore gerontology into daily lifestyle interventions.
Yes, let's look at the interventions that are accessible right now and how they actually manipulate the biological mechanisms we've just discussed. Because you don't necessarily need a gene therapy to alter your aging trajectory.
And the number one intervention, without a doubt, is the one nobody wants to hear because it requires actual physical effort.
It's exercise.
Yeah. It's not a magic pill, it's just sweat. But I want to understand why it works on a cellular level, not just cardio is good for your heart.
Exercise remains the most robust, consistently beneficial intervention for healthy aging across virtually every single biological measure we have. Let's look at high-intensity interval training, or HIIT.
Okay.
When you push your body to its cardiovascular limit, you deplete the energy reserves in your cells. This activates an energy-sensing enzyme called AMPK.
And why is AMPK important?
Because AMPK directly antagonizes the mTOR pathway. It tells the cell, we are low on energy, stop growing, and start conserving.
Oh, so intense exercise actually mimics the biological benefits of caloric restriction.
Exactly. It shifts the cell into a protective house-cleaning state. Furthermore, high-intensity exercise stimulates mitochondrial biogenesis.
Building new power plants.
Yes, as we discussed, mitochondrial dysfunction is a primary hallmark of aging. They get leaky and inefficient. Vigorous exercise literally forces your cells to clear out the old, broken mitochondria via autophagy and synthesize brand new, young, highly functional ones.
So cardio upgrades the cellular power grid. But gerontologists seem to be shifting their focus lately. We always talk about cardio, but what about lifting weights? Resistance training seems to be getting a lot more attention in the longevity space.
It is absolutely crucial, perhaps even more so than cardio, as you enter older age. Resistance training directly combats sarcopenia, which is the age-related loss of skeletal muscle mass and strength.
But isn't losing muscle just a vanity issue? Why does that affect how long I live?
Because muscle is not just for moving your skeleton. Skeletal muscle is your body's largest metabolic organ. It acts as a massive sink for glucose.
Sink for glucose.
When you eat carbohydrates, insulin shuttles that glucose into your muscles for storage. As you age and lose muscle mass, you lose that sink. The glucose stays in your bloodstream, leading to insulin resistance, type 2 diabetes, and systemic inflammation.
And that inflammation accelerates every other hallmark of aging.
Precisely. Lifting heavy weights maintains that metabolic sink and keeps your insulin sensitivity sharp.
Okay, so we have to lift heavy things and get our heart rate up. What about the diet side? If cutting calories by 30% is too miserable, what is the pragmatic alternative? This brings us to time-restricted eating, or TRE, which is everywhere right now.
Yes. Time-restricted eating involves confining all your caloric intake to a specific window of time, usually 8 to 12 hours a day, and fasting for the remainder without necessarily reducing your total daily calories.
So I eat the exact same amount of food, I just compress the window. Does that actually work biologically, or is it just a weight loss trick?
The human clinical data is still developing compared to the mouse models, but the biological rationale is very strong. When you eat continuously from the moment you wake up until you go to sleep, your insulin levels are constantly elevated and your mTOR pathways are constantly engaged in growth mode.
This startup company never has a recession, so it never cleans house.
Exactly. By implementing a daily fasting window of 14 or 16 hours, you allow insulin levels to drop to baseline. This periodic drop down-regulates mTOR and provides a daily window for mild autophagy to occur.
That makes a lot of sense.
It also helps align your metabolic processes with your circadian rhythms. It's a pragmatic way to get a mild dose of the cellular house cleaning we see in caloric restriction without the psychological torture of actual starvation.
It's a lifestyle compromise. I can skip breakfast to get a little autophagy. And what about the other massive trend right now? Heat and cold exposure. Feels like every podcaster on the planet is currently sitting in a sauna and then immediately jumping into a tub of freezing water in their backyard.
Yeah, that's definitely a trend.
Is there real longevity science behind this, or is it just biohacker theater?
It's a fascinating area of biology based on the concept of hormesis.
Hormesis.
Hormesis is the idea that a mild, acute biological stressor triggers a protective cellular response that leaves the organism stronger than it was before the stress. What doesn't kill you makes you stronger, applied at the molecular level.
So when I sit in a 200-degree sauna, I am stressing my cells out.
Yes, the acute heat stress causes your cells to express high levels of heat shock proteins.
What do those do?
Remember our discussion on the loss of proteostasis, proteins misfolding and clumping together.
Right.
Heat shock proteins are chaperone proteins. Their specific job is to grab misfolded proteins and help resolve them into their correct, functional three-dimensional shapes. So heat exposure directly activates the proteostasis network.
Oh, wow. And cold plunges.
Cold plunges operate on a similar hormetic principle. The acute cold shock stimulates the production of brown adipose tissue, which burns energy to create heat, and strongly activates that AMPK energy sensor we discussed.
Okay, so the cellular mechanisms make sense, but does it actually translate to living longer? Should I be buying a chest freezer for my garage?
We have to be measured with the data here. There is very strong epidemiological data from Finland, where sauna use is deeply embedded in the culture.
The Finns love their saunas.
They do. Studies tracking tens of thousands of Finns show that frequent sauna use 4 to 7 times a week is highly correlated with a massive reduction in cardiovascular mortality and all-cause mortality.
So their hearts are incredibly healthy.
They are. However, correlation is not causation. And more importantly, the specific impact of hot and cold exposure on pushing maximum human lifespan, getting people past 100 or 110, is still highly preliminary.
It's not proven yet.
It is nowhere near the level of irrefutable biological evidence we have for exercise. Saunas and cold plunges are fantastic tools for cardiovascular resilience and vascular health, but whether they are true longevity interventions that extend maximum lifespan remains to be proven in clinical trials.
Okay, that is a very fair assessment. So let's play this whole thing out. Let's imagine the timeline.
Okay.
You hit the gym and do your high-intensity intervals to boost your mitochondria. You lift weights to fight sarcopenia. You restrict your eating window to trigger daily autophagy. You sit in a sauna to refold your protein.
Doing all the right things.
And let's say 15 years from now, the science delivers on its lab promises.
Yeah.
You get a prescription for a safe, intermittent mTOR inhibitor. You take a targeted senolytic once a year to clear out your zombie cells. And maybe eventually you get a localized epigenetic tune-up to polish the scratches off your cellular CD.
The ultimate maintenance routine.
You are chronologically 150 years old, but biologically you are functioning like a healthy 35-year-old.
Given the current trajectory of the science, that is a biologically plausible scenario within the coming decades.
Which brings us to the ultimate question, the elephant in the room that we cannot ignore. If we can achieve this, if we can cure aging, should we?
The ethics of life extension. This is where biology crashes headfirst into philosophy. And we have to engage with these questions seriously because they are no longer abstract thought experiments for science fiction novels. The technology is arriving.
Let's start with the skeptics, the people who think radical life extension is a terrible idea for the human soul. There is a very famous philosophical essay from 1973 by a British philosopher named Bernard Williams called The Macropolis Case.
Yes. Williams bases his essay on a play about a woman named Elena Macropolis, who takes an elixir of life and lives for over 300 years.
What was his argument?
Williams argued that immortality, or even just indefinite life extension, would ultimately be a nightmare of crushing boredom and apathy. To explain why, he introduced the concept of categorical desires.
Categorical desires. Break that down for me.
Categorical desires are the fundamental, deeply held goals and projects that give you a reason to wake up in the morning. Writing a novel, raising a child, building a business, mastering a musical instrument, seeking justice.
Okay.
Williams argued that these desires are intimately, inextricably tied to our finitude. We have a sense of passion and urgency because we implicitly know that our time is limited.
Right. If I know I have exactly 500 years to write my novel, why would I ever sit down and write it today? I'll just watch TV for the next decade. There's no deadline.
Exactly. But it goes deeper than just procrastination. Williams argued that over 300, 400, 500 years, a human being would eventually exhaust all of their categorical desires.
You'd run out of things to care about.
You would have done everything. You would have seen every landscape, mastered every hobby, loved, lost, built, and destroyed. Your character, your personality would eventually crystallize and become utterly rigid. Life would become an endless, meaningless repetition. He argued that death isn't a glitch in the system. It is the very boundary that gives our choices meaning.
That is a profoundly poetic and somewhat melancholic way to look at mortality. It gives death a purpose. And the bioethicist Leon Kass has a very similar view, right?
Kass argues that the acceptance of our mortality is essential to living well and living virtuously. He views the transhumanist push to transcend our biological limits as a failure to appreciate the beauty of the finite human condition.
So, struggling against death distracts us.
He suggests that struggling against death with such technological fervor distracts us from finding meaning in the life we currently have. It's an obsession with the future that destroys the present.
I understand that perspective. It's romantic. But I have to say, when I hear those philosophical arguments about the beauty of finitude, I immediately think of the other side of the debate.
The advocates.
People like Aubrey de Grey, the founder of the SCNS Research Foundation, and the prominent advocates for life extension, I imagine they hear arguments about the beauty of death, and they are absolutely furious.
They view it as a massive societal scale rationalization, an extreme form of Stockholm Syndrome.
Exactly. Aubrey de Grey argues that aging is simply a disease, full stop. It is a biological accumulation of molecular and cellular damage.
Right.
We treat cancer, we treat heart disease, we treat pediatric leukemia. Why on earth would we draw a line in the sand and refuse to treat the root cause of all of them?
De Grey points out the staggering, visceral statistics. Approximately 100,000 people die every single day from aging-related causes. It is a daily tsunami of suffering and death.
100,000. Every day. It brings us back to our opening hook. If a virus did that, we'd stop the world to cure it.
But because it's aging, we just accept it.
We call it natural. De Grey argues that this fatalism is just a psychological coping mechanism. Throughout history, we couldn't do anything about aging, so we invented philosophies and religions to tell ourselves that aging and death were actually good and beautiful things.
The advocates argue that now that we are beginning to understand the mechanisms, the hallmarks, the senescent cells, the epigenetics, refusing to develop these treatments because of philosophical concerns about boredom is monstrously unethical.
It's like refusing to treat a child with tuberculosis because death is a natural part of life.
That is exactly the moral equivalence they draw.
It is a brutal counterargument. And honestly, it's incredibly hard to disagree with when you frame it purely as alleviating human suffering. Nobody wants to watch their parents lose their minds to Alzheimer's. Nobody wants to lose their own physical independence and end up in a nursing home. If we have the science to stop that, don't we have a moral imperative to use it?
We likely do, but there is another ethical dimension that is perhaps even more pressing than the abstract philosophical debate about boredom, and that is the very tangible societal issue of equity.
Yes. This is where I strongly push back on the techno-utopians who think this will just magically fix humanity. Imagine a world where David Sinclair's epigenetic reprogramming is perfected.
Go.
You can go into a clinic, get a viral vector infusion, and reset your biological clock to 25. But the treatment costs $10 million.
A highly likely scenario in the early decades of any such complex genetic breakthrough.
What happens to humanity then? We already live in a world with massive wealth inequality. But imagine longevity inequality. Imagine a world where the ultra-wealthy don't just have better houses, better food, and better cars, but they actually get to live for 300 years, while the middle class and the poor still succumb to heart disease and cancer at 80.
It would create a biological caste system that would dwarf any economic or social disparity we have ever seen in human history.
That's terrifying.
Think about the compounding effect of wealth over a 300-year lifespan. You could accumulate resources on a scale that is currently impossible. Dictators or autocratic billionaires could consolidate and hold power for centuries. The societal stagnation could be absolute.
It's literally the plot of a dystopian sci-fi movie.
It raises a vital, urgent question for society today, right now, as this research is being funded. Should we be prioritizing the pure speed of discovery, racing toward longevity escape velocity, regardless of the cost of the therapies?
Or should research funding be heavily directed toward interventions that can be cheaply and equitably distributed from day one?
Finding a cheap, generic pill like a Rapalog rather than an elite million-dollar bespoke gene therapy.
And as you said, these aren't just thought experiments anymore. The science is moving incredibly fast. We are the generations, us talking right now and you listening to this deep dive, who are actually going to have to answer these questions. The technology is going to arrive, and we have to decide what kind of society we want to drop it into.
The vocabulary of this field is no longer confined to dense academic journals. Terms like health span, cellular senescence, autophagy, and epigenetic reprogramming are rapidly entering the cultural lexicon.
They are everywhere.
They're part of biotech investment discourse, mainstream journalism, and everyday wellness conversations. Understanding the precise science behind this vocabulary is essential for navigating the future of medicine.
It's a brave new world. We have covered a massive amount of ground today. We started with evolution's blind spot, how natural selection installed that turbo boost of early fitness that eventually burns out our biological engine. We mapped out the specific hallmarks of how we decay, from the fraying of our telomeres to the misfolding of our proteins.
We explored the nutrient-sensing pathways of caloric restriction and how we might hack them with drugs like rapamycin to trigger deep cellular house cleaning.
We talked about hunting down those toxic inflammatory zombie cells with targeted senolytics. But before we sign off, I want to leave you, the listener, with one final, totally provocative thought to mull over. Something that builds on the ethics we just discussed, but goes beyond the biology of the body and straight into the mind.
I'm intrigued. What is the question?
Let's assume the biology is completely solved. The senolytics work perfectly, the epigenetics are flawlessly reset, your body is permanently, biologically 30 years old. What happens to your psychological capacity? Does the physical human brain have a hard drive limit?
That is a profound and completely unanswered question in neuroscience.
Think about it. Our brains evolved over millions of years to hold roughly 80 to 100 years of memories, relationships, skills, and trauma. If you live for 500 years, will you literally run out of synaptic storage space?
It's a staggering possibility.
Will you reach a point at year 200 where, in order to learn a new language or remember the name of a new friend, you are forced to start intentionally deleting the memories of your first 100 years? If you have to delete the memory of your first love or the childhood of your great-great-grandchildren just to stay sane and make room, are you even the same person anymore?
We are pouring billions into solving the hardware, but we have absolutely no idea what an indefinite lifespan will do to the software of human identity.
Exactly. When you look in the mirror tomorrow morning and you see that ticking clock we talked about at the very beginning, just ask yourself, if the clock stopped, who would you become? Thank you for joining us on this deep dive. Keep questioning the consensus, keep learning about your biology, and think deeply about what you want to do with your own expanding health span.